In multicellular invertebrates, the integument is made up of three layers. It could be epidermis, a basement membrane, and a thin layer of connective tissue. So epidermis is a single layer of columnar epithelial cells. We know in animals there are four types of tissues. They are epithelial tissue, connective tissue, muscular tissue, and the nervous tissue. So epithelial tissue is making all the coverings of the body, whether they are exposed to internal environment or external environment. So these epithelium, they are also providing glandular cells. They are making unicellular or multicellular glands. It means epithelium is providing mainly two function. It is making sheets on all the surfaces, covering on all the surfaces, and then it is also making glands or glandular cells. So epithelium, how we differentiate epithelium from other types of cells, that epithelium is having distinct type of cell shapes when we see under microscope. So when we have a section of any tissue from animals, we see whether the cell shapes, they are distinct. They could be cuboidal shapes, mean all the dimensions of the cells would be same, and could be columnar cells in which the height of the cell would be more than the breadth and the width of the cells. And then there could be flat cells, leaf like cells, they are called as squamous. So there are three types of epithelium on the basis of their cell shape. They could be cuboidal, they could be columnar, they could be squamous. Here we can see these are the columnar cells. So these cells, they could be in a single layer, they could be in multi-layer. If they are in single layer, we say it is a simple epithelium. So in case of simple epithelium, it could be a simple columnar epithelium, it could be a simple cuboidal epithelium, or it could be a simple squamous epithelium. And there could be multiple layer. In that case, we say it is a stratified epithelium, that is in the form of strata. So the epithelium, it could be named as stratified cuboidal, it could be stratified columnar, it could be a stratified squamous epithelium. So here we can see this columnar is mostly a simple. So here we can see a simple. So these epithelial tissue, it has another characteristic, that they don't have any blood vessels in them, or there are few nerves in them. So these epithelial cell, because it is living tissue, so it is mostly have connective tissue beneath that layers of epithelial cells. So this connective tissue, it means connective tissue means it is a tissue that connects to different types of tissues. For example, here the connective tissue is connecting these epithelial tissue with muscles beneath. So this connective tissue, it has a lot of blood vessel, in contrast to epithelium, they don't have any blood vessels, but the connective tissue, it is highly vascularized, it has a lot of blood vessel in it. So these blood vessels, they nourish connective tissue, as well as they nourish these epithelial layers. So the when there would be an epithelial layer, then or a tissue, then beneath there would be a connective tissue. This connective tissue is carrying blood vessels, so this connective tissue would be there. And then there is a basement membrane. Membrane is actually a thin layer of extracellular material that is secreted by same epithelial cells and the connective tissue below. So these basement membrane is acting as a scotch tape like structure that is attaching cells to the at the base. So it is an extracellular material, it is released by epithelial cells. So in case of integument in multicellular organism, epidermis is made up of a single layer of columnar epithelial cells, and then there could be a thin layer of extracellular material that is secreted by same cells, and it is acting as a scotch tape like structure that is attaching the cell to the connective tissue. are at the base, and then there is a thin layer of connective tissue. This connective tissue is nourishing this epithelium and attaching it to the muscle layer below. So epidermal cell, they may have cilia. So in body, when we see there are extension of cell membrane, wherever they are, for example, they are in a respiratory tract, there are cilia on the cells, and then there could be cilia on the cells that are present on the inner layer of reproductive tract, or there could be flagella like on sperms or some other tissues in the some like in protozoan. So these all are actually in the cilia, flagella, or microvilli, for example. We know it is another type of extension. So we would summarize that there are about three types of extension that are present on the cells. There could be flagella that are longer, they are few in number. There could be cilia, they are like flagella, but they are more in number, they are shorter, and these both are supported by microtubules from the cytoskeleton of the cell. But the third one, that is microvilli, they are the extension of the cell membrane that are provided by actin filaments that are microfilaments, they support them. So these microvilli, they are the third structure that is extension of cell membrane. So these extension, they are always present on epithelial tissue. They are not present on connective tissue, muscles, or any other type of tissue. So cilia and flagella, they are present on the epidermal cell because they are epithelial in nature. So there could be cilia present on these integumentary cells, epidermal cell. Then epidermis may have glandular cells. We previously discussed that epithelial tissue, it is providing almost all glands. We can count the exception that are from connective tissue or some other nervous tissue, but rest of all the glands, they are from epithelial, whether they are endocrine or they are exocrine glands. As we said that the multicellular organisms, they may have uh, an integument of three layers: epidermis, basement membrane, and connective tissue. The Some of these multicellular animals, they may have an extra layer that is present on the top of their and epidermal cells, and this layer is called as cuticle. It is a non-living uh, surface, and it is secreted by same epidermal cells. So these epidermal cells, they secrete this cuticle and extra layer. So the structure of this cuticle is highly variable. It could be thin and elastic, or it could be thick and rigid, and it could support the body, like in crustacean and arachnids and insects. It is an The cuticle is making an exoskeleton, it is supporting the body. So this cuticle, it could be soft, it could be thin or elastic. When the composition of the cuticle is different than the cuticle that is thick and hard. But the generally cuticles are consist of chitin, a polysaccharide. Like the polysaccharides we know are starch and the glycogen and the cellulose. So these polysaccharide, they are made up of monomers. Here on the top we can see the monomers of the cellulose. So they are glucose molecules. Similarly, in cuticles, the chitin, it is made up of, it is a polysaccharide, it is made up of monomers or monosaccharides. But these monosaccharides, they are different than the glucose monosaccharide, because the chitin monosaccharides, they contain nitrogen in them, so they could be called as amides. So these are the chains of amides. So in chitin, it is a polysaccharide made up of monosaccharides that are having nitrogen in them. And then in cuticles, there would be fibers of proteins. So cuticles are made up of chitin, that is a polysaccharide, and protein. So these make an extra layer on the top of the epidermal layer of the cells. The animals that have cuticle on their surface, mostly the hard cuticle that is present in insects or arachnids. So these animal, when they have to grow, they have this disadvantage that they first have to shed the cuticle and then grow and then again make this cuticle. That process is called as the molting or ecdysis. So in multicellular organisms, the first phylum, that is Porifera sponges, that are the marine animals, they have two layers of cells, and in between these two layers of cell there is a non living material called mesoglia. The outermost layer of cells is called as epidermis, and it is actually the integument, and beneath the epidermis there would be a basement membrane, and then toward inside to because they are vase like animals, toward inner side there are cells that are toward the spongiocele or the cavity of the sponges. So these two layers of cell, that is the epidermal cell, they are called as pinacocyte. Pinacocytes are actually epithelial like cells. They make the integument, and toward inner side are the cells that are choanocytes. These choanocytes, they are collar cell, they have flagella in them, and they move the water out of the sponge. In case of cnidarians, hydra are the example. Epidermis of the hydra, that is the integument, is few layer thick, and the corals, they have this epidermal layer with the modified cell that release calcium carbonate. So these calcium carbonate, they make shells of the corals. So in case of porifera, it is the pinacocyte, the outer epidermal layer, that is making integument, and in hydra there is epidermis, that is few cell layer thick, and in case of corals, this epidermis, it may have modified cells, glandular cells, that release calcium carbonate. So the corals, they make shells. When we go to the phylum Platyhelminthes, that have the tapeworm and the flatworm, these animals, they have a definite tegument or integument, that is called as tegument. Tegument mean to cover. So their integument is made up of same layers, that is the epidermal layer, and then is the basement membrane, and then are the connective tissue, and beneath there are muscles. So this in case of platyhelminthes, that are the uh, flukes and the tapeworm, like here we can see it is like Tinea solemn, and then is the liver fluke. So these animals, they have their epidermal layer in the form of syncytium. So in case of syncytium, the cells, they may not have cellular boundaries, they have multiple nuclei, but the we know in case of cell division there would be karyokinesis first, and then there would be cytokinesis. Karyokinesis mean division of nuclei, and cytokinesis mean division of cells. So there, these are the such type of cells in which karyokinesis have occur, but the cytokinesis would not occur. So these cells, they appear to be in a layer form, that is having only a single cell, that is having only multiple nuclei. So this organization is called as syncytium. So in case of these cells, in case of these animals, the cytoplasm could be divided into two portions, that is the distal cytoplasm and proximal cytoplasm. So in case of distal cytoplasm, it is not having organelles in it, and it is lined by the plasma membrane, and it is having plasma membrane is supported by glycocalyx. Glycocalyx is actually the composition of polysaccharide present on the outer side of the membrane. So the membrane, cell membrane from outer side is supported by the carbohydrate or the polysaccharides, and proximal cytoplasm, the cytoplasm that is near to the nuclei, this cytoplasm is having organelles in it. Distal cytoplasm is actually cytoplasm that is at distance from the nuclei, and proximal cytoplasm is the cytoplasm that is near to the nuclei. So there are two clear portion that is not found in other cells. In other cells, the organelles, they are evenly distributed throughout the cytoplasm, but in case of syncytium of the integument of tapeworm, the epidermal layer, it is in the form of a single cell that is having multiple nuclei, that is called as syncytium, and in that syncytium, or a single cell having multiple nuclei, the outer layer of the cytoplasm that is away from the nuclei is the clear cytoplasm. It is called as distal cytoplasm. It is covered by cell membrane, and that cell membrane is actually supported by carbohydrate from outer side. So we have to remember that carbohydrate, they are always present toward outer side of the membrane. In case of proximal cytoplasm, it is near to the nuclei, and it is having a lot of organelles in it. Then there would be a basal lamina, the third layer, and this basal lamina is actually the composition of the basement membrane plus connective tissue in it. So the basement membrane, we know, is the scotch tape like structure that is, or material that is released by same epithelial cells that is attaching them to the other tissues. So this basal lamina is actually merger of basement membrane and the connective tissue beneath. And then beneath this connective tissue, there are thick layers of muscles. So this type of integument, that is tegument, in the form of syncytium, would help these platyhelminthes, flukes, and the tapeworm to absorb nutrients, and then it is also protecting, because most of these flukes and tapeworms, they are parasites in other animals. So these these integument, it would help them to avoid the. So the tegument is made up of the epidermis, that is in the form of syncytium, then a basal lamina that is made up of basement membrane and the connective tissue, and then are the layers of muscles. So these epidermal cell that is in the form of syncytium, they may have different unique structures on them. These unique structures could be microvilli, or they are called as tubercles. So microvilli, they are also called as microtriches. So microtriches are present in cystodes. These microtriches are fine hair-like filaments that are distributed throughout the surface of the body, and they give the body surface a smooth and silky appearance. They are extension of the cell membrane, so they are coming out of the epidermal cells. So the as cystodes, they are not having any digestive and excretory system. So these microvilli, they act as microvilli in digestive system. They increase the surface area for the absorption of nutrients and elimination of waste material. In fact, the tegument could be called as the gut of animal that is turned out inside out. So because the gut of the animals or the intestine, it is having same type of microvilli. So microvilli are actually, or microtriches are actually the same microvilli. They are supported by the cytoskeleton. We know the cytoskeleton is having actin filaments that are microfilaments and intermediate filaments. So these microvilli, they are supported by those microfilaments. And in case of tubercles, they are called as spines. These are present in trematodes. The tegument Teguments contains a number of invaginations or the pits. So they are externally lined with minute tubercles, among which are dispersed bristle-like projections called spines. Spines are embedded in the basal lamina of the tegument, and the tip is finely pointed to the external surface. So they are also supported or made up of array of actin filaments like in the microvilli, but they are embedded inside the basal lamina. Most part is embedded in the basal lamina, or we can say they are starting from the spines. They are starting from the connective tissue that is beneath the epidermal cells, and their tips are just coming out of the pits that are present in the tegument. And tubercles, they are numerous, they are fairly regularly arranged, and they are rounded. So these two structures could be present on the tegumentation of the platyhelminthes, that is the microvilli, that are extensions. They are also called as microtriches. Microtrix is the singular of these microtriches, and we can see them here. And then there are pits. In these pits there are the spine like structures. So these structures, they are actually, most part of them is embedded in the basal lamina. So these two structures, they are increasing microvilli, they are actually increasing surface area, and then they are helping in the ingestion, for the absorption of nutrients. And the microvilli may increase the surface area for the absorption of the nutrients and removal of the waste. And the spines or tubercles, they help these animals to get stick to the host surfaces in order to get protection and for anchoring into the host tissue. In phylum nematodes and the annelids, they have an extra layer that is present on the outermost layer of the cells, that is the epidermis. So the configuration of the integument is similar to other multicellular organisms, that is the epidermal layer would be present, and in case of nematodes and annelids, these epidermal layers, they could be these are the same epithelial layers. It could be single celled layer, or it could be a syncytium. And syncytium mean the cells, they have multiple nuclei in them, and they are making a single cell that is covering the body. And then there would be a longitudinal muscle that are present beneath these. So it is understood that beneath epidermis there would be a layer of basement membrane, and then there would be a layer of connective tissue, and then are the longitudinal muscle over there. So on the surface of these epidermal layer, there is another layer that is of cuticle. So in case of nematodes and annelids, because they are soft bodied, so their cuticle is not having polysaccharides in them, that are the chitin. Their cuticle is having most of the protein fibers that are collagenous fiber. Collagen is a protein, so these collagen fibers, they are laid down by these epidermal cells on their surface to make a multi-layered cuticle in order to provide protection. So these cuticles, they may also help to make the body stiff when there would be a pressure of fluids inside body in case of nematodes, because they are not having circular muscle, they are only having longitudinal muscle in them. So for hydrostatic skeleton, the uh, cuticle, it acts as circular muscle to produce the stiffness in the body of these nematodes. In case of annelids, the annelids have two types of muscles. They have longitudinal muscles as well as the circular muscle. So their longitudinal muscle, they are helping in the body to proceed forward, to get shorten or to get lengthen. But the circular muscle, they would produce hydrostatic pressure in the different segments of the body for the local. In case of mollusks, the same configuration again, that epidermis is present, and this epidermis is actually delicate and soft, and beneath this epidermis there would be a layer of basement membrane, and then is the connective tissue. And these epidermal layers or epidermal cell, they may have modified cells in them that are glandular cells. They secrete calcium carbonate that make shells on these molluscan animals. Echinoderms, they also have an integument of a thin ciliated epidermis, that their epidermis is having cilia on them, and then there would be an underlying connective tissue layer after basement membrane, and this connective tissue is actually called as dermis, and this dermis contains calcium carbonate in it. So they are providing a skeleton type structure to these kind. The integument of the arthropod is the most complex of the invertebrate integument, because it also provides the exoskeleton to these animals. The arthropod integument consists of a single layered cuticle, that is the outermost layer, that is non-living layer of the integument, and it is having two layers in it, that is the epicuticle, the uppermost cuticle, and then is the procuticle, that is the cuticle present between epidermis and epicuticle. So this cuticle is the epicuticle. It is thin, it is made up of proteins, fibers, and then the lipid layer, it is present on the surface of these epicuticle. And these lipid layers, they help to prevent desiccation of the animal. And the procuticle, it is the inner cuticle, it is thick, it is having the lamella or layers of proteins, and then the chitin is also included in it. We know the chitin is a polysaccharide. It is having monomers that are amide containing nitrogen in them. They are like other polysaccharide, but they have their monomers, they have nitrogen in them. So these procuticle, they are thick, they have protein as well as the chitin, and it could be called as true cuticle. The epicuticle, it is only made up of protein fibers, and then on the surface of these protein fibers, or the layers of protein fibers, there are layers of lipid. The beneath them there is epidermis, and this epidermis is actually secreted this cuticle for its protection. So this epidermis, it is epithelial tissue, and it could be precisely in case of insects, it is called as hypodermis. And beneath this hypodermis or epidermis, there would be a basement membrane, that is the non-living material secreted by these cells to get attached to the surface. And the lipid, that is, as I already told, the lipid present on the epicuticle, it helps as a. The cuticle of the arthropods is hardened by mainly two processes that are calcification and the sclerotization. Calcification is the hardening of cuticle by release of calcium carbonate that is released from these same epidermal cell. They absorb calcium from the hemolymph and then make it into calcium carbonate. They release it on the surface of their procuticle, that is the innermost layer of the cuticle, and it would hard the cuticle like in crabs and lobsters. And in case of sclerotization, like in insects, the cuticle is hardened by the release of special type of protein. In this type of protein, different layers of the protein of the cuticle, these layers are lamella, they get attached to each other through cross linkages. So the protein fiber that are present in one layer, they are linked to the protein fibers that are present in the other layers of the cuticle, so they are in this process get hardened. Then there is another process that is called as the tanning. So if the cross linkages, it is also involving some pigment like the quinone. One of them is the melanin. So quinone are actually the oxidized type of phenols. So these give colors to the cuticle. So in case of insects and in case of the other arthropods, there could be pigments that are present in the sclerized cuticle, or it could be present in the calcified cuticles. So the sclerotization is actually formation of highly resistant and insoluble protein, sclerotin. Sclerotin is actually the protein that are present in the form of layers. So these fibers, they of different layers, they are linked together through the linkages of other fibers. These sclerotoid proteins are the cuticles. They are mostly present in the insects on their mouth parts by which they could chew the food material. So they are present in the hard parts of the exoskeleton and the body. It means composition of the exoskeleton would change as the function of the organs would change. So the exoskeleton, it may be made up of simple cuticle, it could be made up of calcified cuticle, it could have sclerotin in certain parts of the body. The vertebrates includes five classes. We know the fish, amphibians, reptiles, birds, and mammals. We have to see their integument, and their integument composition is similar to the invertebrate multicellular organisms, but they have some different structures or derivative structures that are associated with their integument. If we see fish, fish are having three groups, that are the jawless fishes, cartilaginous fishes, and the bony fishes. The jawless fishes are the most primitive fishes, and the bony fishes, they have they are the modern fish. The jawless fishes, they are lampreys and hagfish, for example. So these lampreys and hagfishes, they have relatively thick skin that is made up of the stratified epithelial cells, and these epithelial cells, few of them are glandular cell. So they secrete a protective layer on the their surface. That protective layer is actually cuticle. So their cuticle is made up of protein fibers that are collagenous fibers. And in hagfishes, these multicellular epidermis, it is releasing slime because they are converted into slime glands. So these slime, they cover the it is made up of mucus, and we know the mucus is actually the polysaccharides that absorb water and give the slippery condition, or the gum like or jelly like condition, or slimy condition on the surface of the body. So this slime, it protects the hagfishes from external parasite, and it gives them name, that is the slime eel. So hagfishes, they are called as slime eel due to presence of a slimy cuticle on their surface. And in both jawless fishes, that is lamprey and hagfish, the outermost layer that we said is cuticle. In case of the lamprey, it is only made up of proteins but not having any secretion on them, that is the slime. In case of hagfishes, the cuticle is made up of protein, and then the slime is also present, that is the mucus, it is polysaccharide. And beneath them, the cuticle, there is a multi-layered epidermis, and beneath that epidermis there is a dead material, layer of dead material that is secreted from this epidermis. And this material is, or layer is called as basement membrane, and beneath that basement membrane there is presence of the connective tissue that is attaching this epidermal layer to the other tissues that are beneath. So this connective tissue, it is also called as dermis in case of the vertebrate animals. And we know the connective tissue, it is highly vascular, and epidermal cell or epithelial cell, they are not having any muscles. So these connective tissue, they are attaching these epidermal cell to other tissues. They are also, connective tissues are also nourishing these epidermal. The cartilaginous fishes, because they have cartilage in their skeleton, and they are not having bones like sharks and rays. They have multiple layer thick epidermis that is made up of epithelial tissue cell layers, and beneath these epithelial layers there would be, we know, basement membrane, and then are the layers of connective tissue. This layer of connective tissue is called as dermis, and inside this dermis grow palacoid scales. Palacoid mean they are tooth shaped, they are like vertebrate tooth. And these palacoid scale, they are also called as dermal denticles. They grow from the dermis, that is connective tissue, and then they appear from epidermis. So palacoid scales are like the vertebrate tooth, and they have a central pulp cavity like in teeth, supplied with blood vessels, and they are surrounded by a conical type of layer of dentine. Dentine is actually cartilage that is hardened by deposition of the calcium. And then the outermost layer of this tooth is actually inorganic substance like calcium, and it is making enamel. So this is like the vertebrate tooth. Palacoid scales, they cannot grow in size, but their number could be increased when the fish would increase in size. So most part of the skin of the cartilaginous fishes, that are sharks or rays, they are covered by palacoid scale or teeth like scales. So it is assumed that these oral teeth, they evolved from these dermal teeth, that are the denticles or palacoid scales, and they migrate into the mouth. But some people say it is other way around, that the oral teeth, they come on the surface of the fish. So it is not still known that whether they are the dermal dentine or the dermal scales, they are evolved from oral teeth, or oral teeth were evolved from the dermal scales. These scales, they give cartilaginous fishes a sandpaper like texture. They protect the fish, and they uh, provide it the streamlining. Also, the bony fish, they have the skeleton of bones, so they are called as bony fishes. They are the most modern fishes. They have same organization or composition of their skin, that is the outermost layer is epidermal layer, that is made up of epithelial tissues. These epithelial layer, they are very thin and we can hardly distinguish it from the connective tissue that is present beneath that epithelial tissue. So the connective tissue present below epidermis is called as dermis, and this dermis, it contains scales in it. These scales, they are in bony fishes, they are different than the pelacoid scales that are present in sharks and rays, because pelacoid scale, they are teeth like, they erupt out from the or they penetrate their epidermis. But in case of the bony fishes, the scales, they are actually bony but they are present inside the epidermis. Their inside the dermis, their epidermis remain intact, so the scales, they would not come out from the epidermis. So these scales, we have seen them in our practical classes. They could be ganoid scale, they could be cosmoid scale, they could be teleos scale. These scale, they could be cycloid, they could be ctenoid scales. So these scale, they all are having a bony structure and then the epidermal layer covering these bony scales. And the skin or the epidermis, it is also having, that is the outermost layer, it is also having the mucus glands in it, so they release mucus. Mucus is, we know, slimy polysaccharides that absorb water to make a gel-like structure, and it prevents the fishes from bacterial and fungal infections and reduce the friction also. So there could be granular glands that are present on the skin that release the poisonous alkaloids. These granular glands, they are actually modified epithelial cells. So the teleosts, they may that live in deep aquatic environment, they also have modified epithelial cells that are called as photophores. They radiate, they shine in the deep environment. Therefore, difference in the cartilaginous fishes and bony fishes is that the cartilaginous fishes, their dermal scale, that are pelacoid scale, they are teeth-like, they penetrate epidermis, while in case of bony fishes, these scales, they remain inside the dermis, and on them there would be a thin layer of. The scales of the fishes, they are same structure to that of the hair in mammals. Both these structures, they have similar genes that are responsible for the formation of scales and hairs. So these because these both structure, they are dermal in nature, they originate from the connective tissue that is present beneath the epithelial tissue. So these uh, uh, scales, they are generally made up of collagenous protein fibers that are covered by thin flexible layer of bone. We know there are pelacoid scales that are here in the diagram A. Then we have cosmoid scale, and we have the ganoid scale, and then we have teleos scales. The pelacoid scales, they are teeth like, they are only present in sharks and rays, and these pelacoid scale, they have a pulp cavity, the central cavity, and it is having the blood vessels and nerves, and on them there is a bony structure that is called as dentine, and then on this dentine the enamel is present. It is enamel is made up of hydroxyapatite, a crystal of calcium and phosphate, that is the hardest structure in universe. So then the uh, these pelacoid scale, they are not present in bony fishes, they are only present in cartilaginous fishes. And in bony fishes, they could be cosmoid scale, they could be ganoid scale, or they could be teleos scale. Teleos scale are the simplest one because they are made up of only bones. So these bones, they bony structure, they get originate from the connective tissue, that is the dermis, and they remain in dermis. They don't penetrate the epidermis like in case of pelacoid scale. So these are only one layer of bone, and then the in case of cosmoid scale there are, and gonoid scale, there are two layers of bones, the vascular bone and the lamellar bone. Lamellar bone is actually compact bone. Compact bone mean the fibers of protein in these bones, they are compactly arranged to each other, there are no spaces, so this is making a layer that is called as lamella. And above them are the vascular bone that are having pores in them. The fibers of the protein, they have gaps between them, so they appear to have pore in them. So in both the gonoid and the cosmoid scale there are two layers of bone. The only difference is presence of dentine on the upper layer of the vascular bone. So lamellar bone is the deepest layer, and then is the vascular bone, and then on top of that is the dentine, and on the dentine there is a layer of enamel in case of cosmoid scale. In case of gonoid scale, it is the deepest most layer is the lamellar bone, and on that is the vascular bone, and on top of that there is enamel, and in between dentine is absent, that is present only in cosmoid scale. So the bony fishes, they may have the cosmoid scale, they may have gonoid scale, but most of the modern fish, they have only teleost scale, that are the cycloid and ctenoid. We know cycloid are circular, and they have the growth rings in them, so they grow with the age of the fish. But in case of pelacoid scale, we saw they don't grow, but their number would grow with the size of the fish. And cycloid scale, we say they are circular. Ctenoid scale, they have the ridges on their posterior side, so they are